Okay, good evening, everyone. And you know what? Good evening to some of you. It's actually still good afternoon to some of you. And it may even be early morning for some of you because we were expecting and saw a few registrations from Europe. And so if you're here, Europe and I believe other parts of the world as well. So if you're here as well, it may be good morning for you, very early morning. My name is Pam Husband, and I am the Executive Director of the Canadian SADS Foundation. And it's my pleasure to host this webinar where this is a new project for us, and we're very excited to be able to be doing this. So before we start, I'd like to set some basic ground rules. The rules are always a way, hopefully, of ensuring the enjoyment of all at this session. And so one of the more important rules, I think, is to note that Dr. Roberts will provide in-depth information on the topic. This webinar is not intended for personal medical advice. And so we encourage you to ask general questions related to today's topic and not necessarily about your own personal health history. To maintain webinar etiquette, we kindly also ask all participants to keep their cameras off during the presentation and microphones muted. This minimizes distractions for everyone. If you have any questions during the presentation, please feel free to type them into the chat. And we will address as many as we can during the Q&A session after Dr. Roberts has completed his presentation, which will last, our Q&A will be 10 or 15 minutes. The presentation is likely to run about 30 minutes. And before we begin, I just do want to introduce Dr. Jason Roberts. Dr. Roberts is a distinguished clinical cardiac electrophysiologist at Hamilton Health Sciences, a PhRI scientist, and an associate professor of medicine at McMaster University. His clinical research has been pivotal in enhancing our understanding of genetic arrhythmia syndromes. Dr. Roberts completed his cardiac electrophysiology training at the University of California, San Francisco, and his cardiology fellowship at the University of Ottawa Heart Institute. He also holds a master's degree in clinical epidemiology and biostatistics from UCSF. We're looking forward to an engaging and informative webinar. Thank you for joining us today. Now, without further ado, I'll hand it over to Dr. Jason Roberts. Thanks, Dr. Roberts.
Excellent. Well, thank you so much, Pam, for the invitation. It's great to be here talking about this topic that is very dear to me and discussing this clinical trial that we're looking to launch in the next few months. So the topic today is, as alluded to, this clinical trial that we're calling the TARGET trial, which is evaluating what we believe is a potential disease-modifying therapy for arrhythmogenic cardiomyopathy, the most common subtype being ARVC. So in terms of overview over the next 30 minutes, I'll just initially I'll provide a bit of brief background about arrhythmogenic cardiomyopathy and ARVC to provide some context for the clinical trial. I suspect this will probably be review for a lot of people, so we won't spend too much time on the individual components. And then we'll move into the TARGET trial. I'll discuss the rationale, how we got here, and then discuss the design of the clinical trial, what it will involve. So in terms of background, so arrhythmogenic cardiomyopathy, what does that mean? So it's a heart muscle disorder associated with a risk of sudden death. When people have weak heart muscles, we call those cardiomyopathy, the risk of sudden death usually is reflective of the how bad the pump function is. So when the left ventricular ejection fraction becomes less than 35% in people that have regular cardiomyopathy or regular non-ischemic cardiomyopathy, that's usually a trigger for us to put in a defibrillator. But arrhythmogenic cardiomyopathy is a subtype wherein the risk of sudden death happens before the pump function gets severely reduced and or is related to the right ventricle. So as alluded to, the most common subtype of arrhythmogenic cardiomyopathy is ARVC, or arrhythmogenic right ventricular cardiomyopathy. And this is a heart disorder wherein the right ventricular muscle gets replaced with fibrosis or scar and fat. And replacement of muscle with scar creates a substrate or a vulnerability to dangerous fast heart rhythms, ventricular tachycardia and ventricular fibrillation, that can result in sudden death. And this can develop in young, otherwise healthy people in their 20s and 30s, and can result in very tragic outcomes. It's considered a rare disorder, but it's not that rare. So the prevalence is estimated at about 1 in 5,000 in most regions. So in Canada, with a population of around 40 million, we anticipate we have just under 10,000 patients with this condition. So how is it diagnosed? There's no one single test that makes a diagnosis of ARVC. It's a constellation of features. They're called the task force criteria that we use. So one of the diagnostic modalities that we use to make the diagnosis is the ECG. On the ECG, the most common finding that we have in ARVC is T-wave inversion. So the T-wave is being flipped. So here is a T-wave in lead 2 being upright. Here it's down. So if it's down in V1 through V3, that's considered a major criteria for ARVC. There's something called an epsilon wave as well. So this is a specific finding. So if it's present, it's highly suggestive of ARVC, but it's not present in the majority of patients. And this is a late deflection after what we call the QRS complex on the ECG. And this is felt to be reflective of significant scarring in the heart, such that the electricity, it goes quickly through the normal muscle, but when it gets to an area of significant scar, it meanders and takes a little while to get through. So it's the activation of that scarred region happens late. So that's how we think the epsilon wave manifests. In addition to the ECG, so imaging, taking pictures of the heart are important for diagnosis as well. So echocardiogram or ultrasound of the heart is important. In ARVC, the right ventricle is often big or dilated. So this is just a picture showing a dilated right ventricle. And MRI is a standard component of imaging in ARVC because echo oftentimes doesn't see the right ventricle quite as well. And MRIs can show aneurysms of the right ventricle. An aneurysm is when the heart muscle squeezes, the aneurysmal part bulges out. So it's a still image. It would be nicer to show a video, but you can see a bulging here in what we call the RVOT, or the right ventricular outflow tract. And here, lower down, you can kind of see crinkling of the right ventricular free wall. We call this the accordion sign. These are kind of a series of aneurysms that are juxtaposed next to one another. So this is an ECG, whereas the ECG I showed before, where the heart rate was slower, this is an ECG of ventricular tachycardia. So this is a dangerous fast heart rhythm that patients with ARVC can get. And this can cause fainting if the heart rate is fast enough and lasts long enough, and potentially can cause sudden death if it persists. As far as why people get ARVC and ACM, so it's a genetic condition. There's multiple genes that are involved. I've listed 6 here. These are the 6 genetic subtypes that are eligible for our target trial. ARVC is often considered a disease of the desmosome. Those are the kind of the portions of cells that help cells stick together, the kind of adhesion portions. So there's 5 desmosomal genes that have been implicated in ARVC. The most common one is PKP2 or plakophilin-2, and then there's 4 others. And then as well, another important genetic subtype, especially here in Canada, is TMEM43. This is a common form of ARVC in Newfoundland. So they actually, the Newfoundland group discovered this gene, and they have hundreds of patients in the province that have this genetic subtype. So in terms of, so those are the genes that are involved. Why the disease develops, so we call it the pathophysiology. So we think that there's a problem with cell adhesion when these genetic variants are present oftentimes. But it's not as simple as just the cells having trouble sticking together. Beyond that, the genetic changes result in more complex things, changes in gene expression, potentially calcium handling. There's a lot of complex, more downstream things that ultimately happen. So how do these patients, or how do ARVC patients do, and how do we manage them? So I'm sure the audience is familiar with this. So if people just have the genetic change, but don't have any clinical evidence of the disease, we generally monitor and usually don't institute medications at that point, assuming clinical testing is normal. But when we start to see clinical changes on either ECG, Holter monitoring, or ultrasound or MRI, then we oftentimes will start a beta blocker. Not all doctors agree with this, but oftentimes we'll start a beta blocker. And as many of you probably know, exercise, especially intense exercise, can accelerate disease progression in ARVC. So we oftentimes tell patients that they have to start to curtail their exercise. How much exercise restriction is necessary? That's also a matter of debate, but certainly high intensity sports, long duration, doing it 7 days a week, well, there's a lot of data to suggest that needs to be avoided, especially after the disease develops. Once the disease develops, the risk of life-threatening arrhythmias or dangerous fast heartbeats becomes an issue that we have to confront. And when we think the risk is high enough, then we recommend insertion of defibrillators. Defibrillators, they're certainly not a panacea here. When people get fast heart rates, the device shocks them. Oftentimes, shocks are painful, so it's important for doctors to work really hard to prevent these shocks, to prevent the fast heart rates. And we use antiarrhythmic drugs for this, as well as potentially catheter ablation. In addition to this, so if the heart muscle gets weaker and weaker, if medications aren't enough to slow it and prevent it, and in some cases, heart transplantations need it. So I alluded to the need to put in a defibrillator, when to decide to put in a defibrillator. There's a risk score that's been developed for ARVC by one of our colleagues in Montreal, Julia Cadron-Torigny, who I think is giving one of these seminars this month and may already have done so. So she has a very nice risk score where we put in multiple clinical features to predict the risk of arrhythmia. So that helps us discuss with patients about timing of the defibrillator. In addition, so to the right side of the heart, so ARVC, there's also forms of arrhythmogenic cardiomyopathy that involve the left side of the heart. And I won't go through this in detail, but there's other genes in addition to the ones that I mentioned that are relevant here. And we have different thresholds for defibrillator in those cases. So I mentioned ablation can be important, can be very helpful for ARVC. Usually with ablation, we go through the veins in the groins and slide catheters to the inside of the heart, the endocardium. ARVC, though, the disease often begins on the outside of the heart, or the epicardium, and grows inward. So in that context, usually these ablations also often require an epicardial approach. So that's where we have to usually put a needle just below the sternum to get access to the pericardial space so that we can map the outside of the heart in addition to the inside of the heart. And ablation, these are long procedures and they're not risk-free, but they can be very effective at reducing arrhythmia burden. So that's a bit of background about ARVC and in ACM, and I suspect many of you are familiar with those details. Up until this point, the medications that have been used for ARVC are largely borrowed from other aspects of cardiology. So this beta blockers are used for heart attacks and heart failure, for example, after heart attacks, and these medications have been borrowed from those other areas of cardiology to be used in ARVC. But ARVC is different than them. It has its own unique disease process, and ideally we have drugs specifically tailored or targeted for that underlying disease process. So, and we hope we have something like that that we'll be evaluating in the target trial. So how did we get to the clinical trial that we're about to be pursuing? So there were a group of scientists and cardiologists and arrhythmia specialists in the United States that previously studied a series of molecules using a fish model of ARVC. What they did, so it was an unbiased screen of a compound bank of 4,500 different compounds that were that a fish model of ARVC was exposed to, and they looked to see whether or not any of these compounds were able to prevent ARVC from developing. I won't go through the technical details here, but essentially this ARVC fish model would develop heart failure. And they looked to see, could any of these compounds help prevent heart failure from developing? And there was one molecule that I'll abbreviate as SB2 that seemed to be able to prevent heart failure. And then when they did a deeper dive into the effects of SB2 on the fish, in addition to preventing heart failure, it prevented the electrical or arrhythmia abnormalities in the ARVC fish. And when they dived deeper into the molecular structure of the fish, it seemed like this compound was preventing all of the abnormalities that are associated with ARVC. A couple of years later, so members of the original group and some others looked at this same molecule, SB2, in two mouse models of ARVC. One was desmoglein-2 and another one was plakoglobin. And they found similar benefits to this compound in the mouse models. So what did they look at in the mouse models? They looked at pump function of the heart. Vehicle means placebo or kind of sugar pill. And SB2, this is the active compound. These gray boxes here are normal mice, and their pump function's normal. The mutant mice that are given the sugar pill, their pump function is reduced. So up here is the normal pump function. This is reduced pump function. If the mice are given the drug, you can see that their pump function is protected. It doesn't go down like the mutant mice that are given the sugar pill. And then if we look at ventricular arrhythmias, so extra beats and runs of extra beats, the white bars here are mice that are given the sugar pill, and the black bars are mice that are given the active drug. And you can see the burden of extra beats is significantly reduced. So that was really exciting. Our team was evaluating the role of a new gene that we think may be a modifier in ARVC. And our collaborators at the Ohio State University, Peter Moller's lab, developed a mouse model of this where this ankyrin gene was knocked out, and they found that this knocking out the ankyrin gene resulted in cardiomyopathy, so weak heart muscle, and arrhythmias. And they asked the astute question whether or not giving this molecule SB2 would potentially be helpful. These mice develop heart failure around 8 to 10 weeks. So here they started giving the mice the drug at 4 weeks when everybody's pump function was normal. And then if you look forward at 12 weeks when they did ultrasounds in the mice, you can see that, and just focus on the 2 columns over here, the mice that were given the sugar pill, the pump function had reduced. The mice that were given the active drug, similar to what was seen in the earlier study, the pump function was protected. And then they asked the additional question whether or not the pump function could be rescued. So here they waited for the mice to develop a weak heart muscle. So at 12 weeks, here you can see the pump functions are reduced. And then after 4 weeks of treatment, you can see that the pump function improved, whereas the sugar pill, the mice that were given the sugar pill, it did not. So I found this whole kind of story very intriguing. We need better therapies for ARVC. So the question was, how do we move this forward to humans? So this compound SB2 has not gone through toxicology studies and is not, we're not able to use it in humans. The safety hasn't been established. So we wanted to see this compound, SB2, it's annotated, it's a class of compounds called glycogen synthase kinase 3 beta inhibitor. I won't go through too many of those details, except to say, we felt in all likelihood this was a class effect. So we asked, was there another GSK3 beta inhibitor that had made it forward to human studies and was safe, had been found to be safe in humans? And sure enough, there is one, a molecule called tidglusib, that it's advanced to human studies. It's been, it was originally evaluated in the setting of neurologic disease, and it did not show benefit there. But it's currently being studied for a condition called myotonic dystrophy, which is not a heart muscle condition, but a skeletal muscle condition. And promising results have been observed for myotonic dystrophy. We decided to reach out to the company that owned the compound that was studying it for myotonic dystrophy and said, you know, we have reason to believe that it could potentially be helpful for ARVC. And they said, interesting. So because tidglusib is different than SB2, it binds to a different aspect of the molecule, and there's you know, certainly its structure is completely different. We thought it was best to first study it in mouse models. So our colleagues at Ohio State have studied it in a couple of mouse models. This is the data for Anchorin B, the Anchorin B mouse model, and the same things that we observe for SB2 are also observed for tidglusib. So this is a prevention study. If the mice are given, so here's mice that are given the sugar pill, their pump function is reduced. Mice that are given the drug, tidglusib, similar to SB2, their pump function is protected. And then from an arrhythmia perspective, the same kind of benefits were also observed. These are just different charts of mice showing the mutant mice develop arrhythmias in response to adrenaline infusions. But if the drug is given to the mice beforehand, their vulnerability to arrhythmias is dramatically reduced. So that's if the drug is given before the disease develops. And then here is, they wait, similar to what I discussed before, here they waited for the mice to develop ARVC or the changes. And then if they give the drug, then and then expose them to adrenaline, again, their vulnerability to arrhythmia is again reduced. So we were really excited about that because you know, tidglusib, we can study it now in humans. So in that context, we're excited to be starting in the next few months a clinical trial that will evaluate tidglusib in ARVC and arrhythmogenic cardiomyopathy patients. The study will involve 17 Canadian sites. I'll show them at the end. And patients will be randomized to tidglusib versus placebo, placebo being essentially the sugar pill. We need to randomize patients to see if it works. If everybody got tidglusib, we'd not be able to know if it was beneficial. In terms of what we're going to look for benefits, we're going to look at a bunch of things, but the main thing is we're going to look at extra beats and on Holter monitoring So patients will have their Holter monitor performed at baseline, then will be treated with drug or placebo for 6 months, and then we'll repeat the Holter monitor to see how many extra beats they're having. ARVC is associated with lots of extra beats as a general rule. So if the drug works, we're very hopeful that the burden of extra beats will go down dramatically over time in the patients that are given the active drug. Who can get enrolled into the study? So it's for adults. If it ends up being successful, I think it certainly would make sense to evaluate in kids subsequently. But to start, we're evaluating adults. The genes that I had talked about, so the 5 desmosomal genes in the TMM43 variant, those are the patients that are eligible for the study. You need to be having lots of extra beats at baseline in order to get into the study. So you'll have a monitor as part of the screening process. And you need to have had a diagnosis of ARVC or ACM for at least 6 months prior to screening or randomization. The reason is because after someone's diagnosed, we know that the burden of extra beats oftentimes goes down anyways with modifications that are done after diagnosis. So we want people to ideally having stable numbers of extra beats. Exclusion criteria, I won't go through this in detail. It mainly has to do with, we just want to make sure that everybody's kidneys and livers are functioning normally at baseline, just in terms of how the drug is metabolized. Yeah, I think I'll leave that from there. Most of these are just in relation to tideglosib. In terms of the hypothesis, so as mentioned, so similar to the mouse studies, the hypothesis is that tideglosib will reduce the burden of extra beats that people have. So that's the main thing that we're looking at. But in addition, we're very hopeful that the heart muscle will improve. So patients will have ultrasounds at baseline and again at 6 months to check how the heart muscle is functioning. The study we don't think is going to be large enough to detect differences in this, but the ultimate hope is that if tideglosib works, it will reduce burdens of the dangerous fast heartbeats, so reduce the need for defibrillator shocks and episodes of ventricular tachycardia, these life-threatening arrhythmias. So that's a brief overview of the trial. Here are the 17 Canadian sites that are involved, so it's really coast to coast. And so we're hoping that patients in Canada that are interested in the study, there'll be a site nearby that will be able to support their involvement. So with that, I think that's about 30 minutes. So I will thank everybody for your time, for listening, and I'm certainly happy to take questions. So I'll hand things back over to Pam at this point.
Yes, thank you very much. That was really great. And so we did collect questions from people who registered, and I can certainly ask those questions orally. But is there anyone in our audience that would like to unmute themselves and ask a question now? And then if we don't cover all of the questions that came through with registration, then I can go through those as well with asking Dr. Roberts.
I have a question, Pam. It's Heather.
Yes, hi Heather. Nice to see
Hi.
you.
Yeah, I'm wondering how this is different from what we're hearing about what's going on in the United States with gene editing. Specifically, there's a Rocket Pharmaceuticals trial that's going on at, I think, in San Diego and at the Philadelphia Children's Hospital. Can you, if you know about that trial, can you tell us how it's different, or if
Yeah, absolutely. So yeah, there's 3 biotechnology companies that are evaluating gene therapy approaches for ARVC. They're looking at the genetic subtype of ARVC caused by the plakophilin 2 gene, or PKP2. So yeah, I think Rocket Pharmaceuticals, Tenaya Therapeutics, and Lexeo are all evaluating PKP2 ARVC gene therapy. So what that involves is a single injection of a viral vector, so adeno-associated virus, AAV, that will deliver a copy of the PKP2 gene into the heart. And patients with PKP2 ARVC, they usually have one copy that doesn't function and one functional copy. So if they're able to deliver a functional copy to the heart, that will replace the non-functional copy. And the hope is that that will potentially serve as a cure. So that's really, really exciting. This is obviously different. So this is a small molecule. It's a powder that you would take daily that we hope, will potentially help protect people that have ARVC, prevent progression of disease, and potentially prevent it. We think that it might work for multiple genetic subtypes. So that's kind of the difference. I think that hopefully all of them work. That would be great. I think for patients. The gene therapy, it's for a particular subtype, and if it ends up being a cure, then I think that PKP2 ARVC will be then taken care of. So this could be used for the other genetic subtypes potentially. And not everybody probably may want gene therapy up front or in the early days. So this might also be an option for those patients. But hopefully everything works. I think it's a very exciting time for therapeutics in ARVC.
So Heather, is that pretty much answering your question?
Yeah, that's great. Thank you very much.
Thank you, Heather. We have a question in chat here. One question is, what is the next step or steps for this research that Dr. Roberts, you've delivered this evening? What comes next after this phase? Presuming that this clinical trial has some positive results, is there step 2 involved here?
Yeah, absolutely. So we have Health Canada approval to move forward with this trial. We got it a couple of weeks ago. So we're going to be working with the 17 sites in Canada to activate them over the next few months. And we're hoping to start enrollment in, I suspect, hopefully around February, March of 2024. We anticipate it will probably take between 6 to 12 months to enroll 120 patients, maybe a little longer. It can sometimes take a little while to enroll patients, and then they need to be treated for 6 months. In this overall context, we suspect it will probably take about 2 years to complete this trial, approximately. Maybe it'll be quicker. It might take a bit longer, but that's the approximate timeline. If we find that it works in this context, the next step would likely be a larger. So if the study shows a lot, promise, it will be exciting. But ultimately, the next step would be a pivotal trial where it would be a larger study looking at harder outcomes, so outcomes that matter to patients. So the next trial would likely be randomizing to the drug versus placebo and looking at defibrillator shocks and dangerous fast heart rates and death, those and cardiac arrest, those bigger outcomes. That study would likely need to involve probably about 500 or 600 patients, and it require a couple of years of follow-up. So this is a first step to get a sense of whether or not the drug is helpful. It's an early first step, but there would likely need to be a larger trial after this. So drug development, the COVID vaccines have been an exception, but drug development after the first study, usually there's another study that needs to be done before you get to clinical approval. So those are our current thoughts now.
Okay, and we have another question in chat too. What is the name of the rare gene you mentioned? And I believe this person is referring to the rare gene that is often associated with the Newfoundland type of ARVC. And is that screened for in the US as well? And I'm presuming they're asking whether it's screened for genetically.
Yeah, so genetic panels, clinical genetic panels, and it do screen for all those genes, or at least the most common ones. So yeah, TMEM43 S358L. So it's a single letter change in this gene. Most of the patients, there was a founder effect. A lot of them are found in Newfoundland. However, after they discovered the gene and people around the world started screening from them, patients have been found all over the world with this particular subtype. So in that context, clinical genetic panels, when someone has ARVC and you do genetic testing, that gene is usually included.
Well, it was TM43 S358L. Is that what it was?
Yeah, TMEM43. That's the gene, TMEM43. That's the gene, TMEM43, and then the variant is S358L.
Thank you.
You're welcome.
Thank you for asking. Okay, another question.
One minute, Pam, just to interject. There's someone who's had their hand up here for a bit.
Oh, okay,
Marco,
thank you.
if
Yes,
you-
Marco, I see that. You'll need to unmute. There you go, Marco.
Yeah, thank you. Thanks. Hey, Dr. Rob, this great presentation. It's so good to see this progressing and, see that public funds are actually funding this clinical trial. So it's quite amazing. So my question is related to GSK3. Do you expect it to be a target, also for gene elusive in general, ARVC? And are there drugs that might be available already, or people are using? And I'm thinking about semaglutide or Ozempic, that might affect GSK3. We know there are some benefits to the heart, so just wondering about that.
Yeah, so thanks very much for your question, Dr. Prado. Yeah, as far as will it work for gene elusive? So I think one important thing to highlight is the mechanism in terms of how the compounds are working. It's still being worked out. We think it is through modulation of the GSK3 beta enzyme. However, to emphasize, like, it was a non-biased screen. So whereas a lot of drugs are developed with rational drug design, the pathway, and you pick a particular target. This was 4,500 compounds to screen in a non-biased way, and this is the one that worked. Since the original paper, there's been a lot of work to try to tease out the mechanism, and I think we're learning about it, and it's felt there is evidence that it is through GSK3 beta modulation. Are there other factors that are also being modulated? I don't think we totally know. As far as what is it doing in the cell, like, there's suggestion that it may normalize localization of beta catenin, and the desmosomes seem to also their integrity seems to be reestablished. But I think we're still learning about that. But because we know the compound is safe, we think it's reasonable to study while the actual mechanisms continue to be worked out. As far as other compounds, like the GLP-1 agonists, it's tough to know, like, as far as, so I don't know, actually, you can probably let me know, but in terms of GLP-1 agonists, do they modulate GSK3 beta activity? So we don't think it's through kind of glycogen metabolism that this is working, even though that's the name of the compound. We think it is through, so GSK3 beta, it's involved in many different pathways. So it got its name originally from its role with glycogen metabolism, but it's involved in many other things. And we wonder, so it's been wondered, is involved in potentially modulation of Wnt beta catenin, or different things. I think as indicated, we're learning, but from a GLP-1 agonist, if you're kind of wondering, because it's named glycogen, I don't know. I think that would probably be a stretch, at this point. I certainly don't have, compelling data to suggest that they would be helpful. Do you want to qualify or comment?
Yeah, no, just, I mean, I've seen a couple of papers suggesting that those drugs can change GSK3 activity, but not in the heart, right? In other tissues.
Sure.
And I was just wondering because, a lot of clinical trials showed a lot of benefit with these drugs, right?
Yes.
Which is probably through the weight loss too, but I was wondering if there is some evidence for direct actions in the heart, so.
Yeah, no, I will have to look that up. I wasn't aware that the GLP-1 agonists have been shown to modulate GSK3 beta, but interesting. That's good for me to know.
Thank
Okay,
you.
you know
It's
what?
really like amazing.
Sorry, I didn't mean to interrupt. I was just going to say, it looks like we have a lot of interest here, so we may have to, if we cut this off, you're welcome. We will follow through with any questions that don't get answered and make sure that we can try and answer them for you after this. But we have a few minutes, I think, as long as Dr. Roberts is all right. Another question here, can this therapy provide any benefit for people without any known genetic mutation for their diagnosis of ARVC?
Yeah, so that's a great question. If it shows benefit in the genetic subtypes of, or the subtypes where a genetic culprit is identified, I think a logical next step would then be to evaluate it in genotype negative ARVC. For this initial trial, because the mouse models that have been studied, we know these certain genetic subtypes, the mice respond, we felt it was most prudent to evaluate humans with those particular genetic subtypes. But if it ends up showing benefit, then I think it definitely would make sense then to subsequently extend subsequent trials to involve genotype negative ARVC. But as I also want to emphasize that just because there's a lot of, it works very well in the mice, we can't guarantee that it's going to benefit humans. That's why we need to do these studies. We really hope that it will. But that's, there's lots of times that things that are observed in mice don't translate to humans, but we're very hopeful that it will. But if it does show benefit in the genetic genotype positive patients, then I think it absolutely makes sense to then evaluate in genotype negative patients.
Great. Okay, another question. I am seeing my EP tomorrow, and can I ask to be enrolled in this trial, or how, in fact, can I enroll in this trial when it's available?
Yeah, so we absolutely, so right now the study is only in Canadian sites. If your site was one of the ones that I listed, so those sites are all will be looking for patients. So we absolutely encourage patients, if they're interested, to bring this up with their doctors. So our team, so members from all of these 17 sites, we have been having regular meetings over the past 6 months planning for the launch of this clinical trial. So once sites are activated, people will begin actively screening for patients. But while we're waiting, and certainly throughout the duration of the trial, if patients hear about this and are interested in being involved, you can certainly reach out to me, and I can connect you with the site PI, the site investigators. But similarly, if you're seeing your doctor, bring up this trial, I think that's great. Like, part of I'm more than happy to be here, but one of the goals of this as well is to let patients know about this. So if they're interested, that they can, express interest to their doctor. I think that'd be great.
Okay, and we can follow up and connect this particular person with you, Dr. Roberts. We'll make sure that we have the email that you're happy to use with patients and so on, and we can follow up after this evening with this. Another question, do you anticipate expanding this trial to any US sites?
Yeah, so we're going to see how enrollment goes within Canada. So I will say that a lot of this work, it actually stemmed from my time in San Francisco as a trainee. So on our steering committee is one of my mentors from UCSF, Dr. Mel Scheinman, and then Peter Moeller's lab is at Ohio State, and they've been incredibly important in all of this. Because the funding is through Canada, we're going to see if we can enroll the planned 120 patients through Canada. But if enrollment is slow, it's certainly possible that we would expand to the United States. If this initial study is positive, if it shows benefit, then the next larger trial, we definitely anticipate that it would involve other countries, and certainly the United States would be one of the first ones that we would go to.
Okay, now there was a question that came through in enrollment. What are the differences in, I think I sent it to you, Dr. Roberts, the differences in AAV vectors, AAV9, etc. What are the differences? I guess what I would ask is, because this person asked the question, is there some significance to the reference to these vectors in terms of your research?
Sure. So the AAV vectors, they're not relevant to to this trial, to Tiglusib. So Tiglusib is a small molecule that you will take. So it's a powder that it's dissolved in liquid and you drink. AAV vectors are relevant for gene therapy. So in order to get the PKP2 gene, that's what is currently being, or the trials that are being launched in the United States this year. The AAV vector is what's getting it into the heart. I am not an expert at AAV vectors. Generally, AAV8 and AAV9 are the ones that are best able to deliver genes to the heart. At least that's what's my understanding. In terms of going into more nuances about AAV vectors, I would say that there's probably other experts that are better able to answer those questions. But the ones that are usually often used for the heart are 8 and 9. I'm honestly not sure what Rocket, Tenaya, and Lexio are using for their programs.
Okay, now I just want to say thank you to all our participants this evening. It looks like we've pretty much covered everybody's questions.